Hey, welcome everyone. Ilana Witten and I will be talking about connectomics as applied to the basal ganglia. I'll speak first for about 10 minutes and then she will come on. Just to give you a bit of a primer, the basal ganglia are a set of phylogenetically old and evolutionarily conserved subcortical nuclei that are necessary for executing coordinated sequences of motor actions. They're also necessary to promote the repetition of behaviors that led to good outcomes in the past. The importance of the basal ganglia to human behavior is made clear from diseases caused by perturbations of the basal ganglia that include Parkinson's, schizophrenia, OCD, and Tourette's. I'm just going to tell you a little bit about the circuitry in order to motivate today's talk. Basal ganglia have an input stage that's known as the striatum. The striatum receives glutamatergic inputs from cortex, thalamus, and many other brain areas. And from the striatum emanate two inhibitory long-range projection pathways known as the direct and the indirect pathways. These are formed from bistriatal projection neurons. These are GABAergic neurons. The direct pathway innervates from the striatum to the substantia nigra reticulata, which in turn inhibits the thalamus. And because there are two inhibitory synapses here, the net effect of activity in the direct pathway is to disinhibit the thalamus, increase activity in cortex, and increase subsequent activity coming into the striatum. The activity of the direct pathway neurons are thought to promote or invigorate movements and also signal that something good has happened. The direct pathway is opposed by the indirect pathway, which makes an additional synapse through the globus pallidus. And because there are three synapses now between the indirect pathway and the thalamus, the net effect of this pathway is to inhibit the thalamus and withdraw excitation from cortex. And the activity of the indirect pathway is thought to inhibit slow or switch movements and to signal that something bad has happened. And this entire signal system is under the tutelage of midbrain dopaminergic centers, which innervate the striatum quite heavily and release dopamine when something good has happened. In ways that are still mysterious, this seems to promote or invigorate movements and also to reinforce that movement to occur in the future. I'm speaking here mostly about the dorsal striatum, which is innervated by substantia nigra compacta, and Ilana will talk more about the nucleus accumbens and its innervation from the ventral tegmental area. Now, there are many models that have been proposed to govern basal ganglia function in action selection. On the left here is the most simplistic model, a so-called go, no-go model. This postulates that the direct pathway promotes movements and the indirect pathway inhibits movements. This model has been very influential and helps explain the symptoms of Parkinson's disease, in which the indirect pathway is thought to be overactive. Nevertheless, this is clearly overly simplistic and incorrect, because we know that both the direct and the indirect pathway are coactive at times of initiation of movements and at the time of termination of movements. More refined models, such as the complementary model shown here in the middle, and I should mention this is from a review from Lexi Kravitz, postulates that the direct pathway does drive movement, but it's the concerted activity of the direct and the indirect pathway acting together within the striatum to select different ensembles of active direct pathway neurons that selects the movement. And lastly, we have other kinds of models, such as the competitive model shown here on the right, which postulates that the direct pathway selects one movement, whereas at the same time, the indirect pathway inhibits all other movements. And so it's the concerted action of the two that suggests that a coordinated single action is selected. In basal ganglia research, there's a long tradition of trying to link structure to function. And so, for example, we believe that there are pathways that link the cortex to the brainstem that course through the basal ganglia that encode different kinds of motor actions, and that the activity of these pathways is necessary or at least modulates selection of particular motor actions. So what can we deduce from macrocircuits, and what kinds of analysis would be benefited by connectomic analysis? Well, one of the most striking features of this circuit is the massive amount of convergence that seems to occur. There are many, many cells in cortex. There are about 10-fold fewer cells within the striatum, and then far fewer cells within globus pallidus and substantia nigra reticulata. In addition, the basal ganglia sensitive areas within thalamus are also quite small. And so there appears to be a great convergence in this pathway. Now, of course, we don't actually know that that's convergence. We don't know that many, many cells project to fewer cells, in the next structure. It could be instead that there's a highly selective pattern of innervation, whereas there are many potential presynaptic partners for each postsynaptic partner. This could be understood better by some connectomic analysis. In addition, what's quite interesting is that the basal ganglia sensitive parts of thalamus actually fan out incredibly when they re-innervate cortex. So there's a huge divergence in that last step. So we have a circuit that goes through a bottleneck in the substantia nigra reticulata. Of course, this is reminiscent of autoencoder networks that are quite popular now in computational neuroscience. And so some structural analysis could help us understand whether this network is set up to perform the kind of dimensionality, reduction, and compression that really occurs in autoencoder networks. Lastly, this projection that goes back from thalamus to cortex is not very well understood. So we don't understand whether this circuit operates in a point-to-point topographically organized manner. So for example, are cells that are active in cortex that lead to activation of the striatum themselves modulated by the feedback that comes from thalamus, or does thalamus target other regions? We don't know the answer to these questions, and connectomics would help. Since the thalamic innervation seems to come back to layer 1, it has the potential to modulate vast aspects of cortex and probably does not meet this kind of point-to-point topography that I suggested earlier. Within the striatum itself, there are also very interesting questions about microcircuitry organization. It's thought that these models that we've presented before about complementary versus competitive action of the direct and indirect pathway are mediated by striatal microcircuits. However, there's a great deal about microcircuitry that we don't really understand in the striatum. So the direct and the indirect pathway neurons are heavily cross-innervated, both within the pathway and across pathways. In addition, there are populations of striatal interneurons that are, although they're present there in just a few % of total cells, seem to be quite important. For example, there are fast spiking parvalbumin positive cells, which like in cortex, ramify extensively within a local neighborhood and can inhibit many other cells. And there are also low threshold spiking, somatostatin positive interneurons that instead send axons to quite distal places and seem to innervate more across large regions of the striatum, while also targeting dendritic subdomains, such as also true in cortex. So in terms of the microcircuitry of the striatum itself, we would like to understand what are the patterns of synapses that exist between and among classes of striatal projection neurons. And we'd like to understand the nature of the interneural connectivity. Interestingly, the striatum only has a very small population of excitatory neurons that occurs in the cholinergic interneurons of the striatum. The rest of the cells are all inhibitory. And therefore, these interneurons need to be engaged by feed-forward drive arriving from cortex, thalamus, and other structures. The nature of recurrent connectivity within the striatum is inhibitory. So if these are excited by striatal activity, it has to be via mechanisms of disinhibition, which would be revealed by particular circuit motifs as seen in connectomics. This microcircuitry analysis will help us understand whether there's a center surround type organization within the striatum, such there's local refinement potentially of actions that are selected. Alternatively, it could be that there's always this long range distant kind of inhibition, which might suggest more sort of cross-modal inhibition in selection of actions. For example, that running might inhibit reaching. Lastly, there could be asymmetry in the patterns of projections that could tell us quite a bit about function. So for example, maybe the direct pathway neurons sit at the top of some kind of inhibitory hierarchy, such that at the same time that one action is selected, automatically other ones are inhibited because those direct pathway neurons have a privileged position in terms of inhibiting other circuit elements. So we would need some connectomic analysis to reveal this kind of microcircuitry. For a variety of reasons, our lab and Bob Dada's lab, with whom we collaborate extensively, prefer to think about some kind of hierarchical hybrid models in which interactions between pathways occurs in the globus pallidus and the substantia nigra reticulata, such that large-scale regions within the striatum that might mediate, for example, running or reaching or grooming, cross-interact within these structures. Whereas within the striatum itself, local circuitry decides within a gross class of actions, specifically which one is selected. For example, what kind of reach occurs. Of course, for this to work and to be able to be reinforced, there would have to be a similar organization of dopamine innervation that would allow selection at the appropriate scale. Now, just to end quickly, there's a couple of other points which are unknown that would be interesting to analyze by connectomics. Buried within the dorsal striatum is a second scale of organization known as the patch and matrix, discovered by Anne Graybill and by Chip Kerfin separately. These consist of molecularly and anatomically distinct striatal projection neurons that are thought to form their own microcircuits, such that the patch is really separated from the matrix. We have very little analysis here about microcircuitry on a global scale for patch matrix organization. This would be really quite important. Lastly, of course, there are whole classes of unknown unknowns to which we don't have access yet. And I wanted to show you one little teaser. This is a very beautiful image created by Jaeon Lee, a graduate student in my lab, that shows a vortex of cells in substantia nigra reticulata. These were labeled by retrograde infection with rabies in a downstream structure. Of course, this immediately suggests maybe the presence of a ring attractor, potentially analogous to those in the Drosophila brain that help mediate orienting behaviors. Microcircuitry might be able to tell us right away whether this actually can function as a ring attractor. That's all I have for you today. I'll just close by saying that I think we should tackle many samples because in order to understand the relationship between circuit activity and behavior, we need to have variability in both. And so analogous to the great insights that came from genotype-phenotype relationships by analysis of many human genomes, we would need to similarly have many connectomes.
My name is Ilana Witten, and I'm an associate professor at Princeton University, and I'll be talking today about the usefulness of the basal ganglia connectome. Midbrain dopamine neurons are a really important part of the basal ganglia, and what I'll be telling you about today is why we think they're so essential to reinforcement learning and how the mouse connectome could further our understanding of the computations that they're performing. Dopamine neurons are known and really are famous for encoding errors in the prediction of reward, which I'll be abbreviating as RPE today. And basically what they do is respond when there are unexpected rewards or unexpected stimuli that predict reward, but not if there's a reward that's fully predicted. And this finding has been incredibly influential because reward prediction error is a critical component of reinforcement learning algorithms. And the way it's calculated is by taking the temporal difference of predicted reward or value, which I'm abbreviating here as V, and then adding it to reward. And this teaching signal has been really, really important for training reinforcement learning algorithms in computer science. For example, recent feats in machine learning, like agents that can play Atari, have been trained on this algorithm, trained with this teaching signal. And there's really a lot of evidence that this neural signal is being used in the brain for reinforcement learning. This comes from many lines of evidence, including optogenetics, where activation or inhibition of dopamine neurons is necessary or sufficient for reinforcement learning. Animals will repeat an action associated with dopamine stimulation and avoid one when it's associated with the inhibition of those neurons. And furthermore, the dopamine release in downstream targets in the basal ganglia is associated with synaptic plasticity, which is thought to be the neural basis of reinforcement learning. So how is this thought to work? What do we think we know about the neural implementation of dopamine-mediated reinforcement learning? This is a classic schematic of a classic hypothesis where the cortex encodes the state of the world, like stimuli in the environment or possible actions or recent actions. And the idea is that these inputs from cortex to the striatum or pallidum in the basal ganglia get strengthened by this teaching signal, this dopamine reward prediction error signal, which is in green in this diagram. And that produces striatal neurons that encode reward prediction or value that's associated with the input neurons because the weights are getting modified by that dopamine signal. And so if you sum all of these striatal neurons, you will get the value or the predicted future reward associated with the state that's being encoded in cortex. And so basically that's really useful because now you'll have striatal neurons that are able to tell you how likely reward is for a given situation. And that is really good for making good decisions based on recent reward or recent reinforcement. So in summary, the idea is that the dopamine signal is converting the state representation in cortex into these value or future reward representations in striatum, and that allows the circuit to predict reward and therefore make good choices. And this is really an elegant framework, but there's at least one major thing that's missing, which is that how is this reward prediction error signal, this really important teaching signal, actually being calculated? And here I've shown it, I haven't shown how is this happening. It's just these arrows, but this is basically the question is really how do you get this temporal derivative of these value inputs that are then summed with reward and converge onto this dopamine signal? And I think that's one thing that the connectome would be really useful for. I'll show you a few different hypotheses, and I think it will be clear that having a multi-region connectome could really distinguish between them. And so one is that the GABA interneurons in the VTA are what are doing this, are really central to this calculation. So you have these value inputs in the striatum that converge in the pallidum to make a total value function. And then you need to take the temporal derivative of that. So you could get that by having a direct input and also an indirect input to the dopamine neurons through GABA interneurons that invert and delay the signal, which is what a derivative is, that get also summed with a reward input to get a prediction error. So that's one hypothesis. Another could be that that temporal derivative, basically, rather than being performed by GABA interneurons in the VTA, that computation goes one synapse earlier in the ventral pallidum, where an interneuron there serves the same function, where you have, again, a delayed and inverted signal through having an extra interneuron pathway through the VP and also a direct pathway. Finally, a third hypothesis is a little bit different, and this has to do with the direct and the indirect pathways in the basal ganglia. The idea is the nucleus accumbens, this ventral part of the striatum, could send a direct pathway input to one class of interneurons in the VTA and indirect through the pallidum to another class of interneurons. And that could also lead to a temporal derivative, because you'll have one pathway that has an extra synapse and an extra negative sign. So that will again delay and invert the signal. So this is a bit of a different hypothesis. And these are really three different ways that are all consistent, at least to some extent, with existing very coarse anatomical data. And I think the connectome could be really, really helpful in distinguishing between these models and therefore leading to an accurate circuit model of reinforcement learning in the brain. And the second thing I want to briefly touch on is how the connectome could help us think about how to reconcile recent understandings that dopamine neurons are actually quite heterogeneous with this classic role of dopamine contributing to reinforcement learning. So in this circuit schematic, there's only a single dopamine neuron, and it would carry a scalar reward prediction error. That's actually at odds with recent data from our lab and also many other labs showing that dopamine neurons are really very heterogeneous under many task conditions. So one link between these new data and this classic idea of dopamine driving reinforcement learning comes from an anatomical hypothesis. And this is work that we're doing with Rachel Lee and Nathaniel Daw, which is to suggest that perhaps different dopamine neurons are all doing the same circuit computation. They're calculating reward prediction based on their inputs, but different dopamine neurons are simply doing that based on different inputs. So they don't have the full state representation that they're using to predict reward. So their individual signals are quite heterogeneous and aren't the total reward, aren't the proper reward prediction error function for the state of the world. However, in one anatomical rendition of this, if all the dopamine neurons were to converge at the target site, that could recover the classic model and get the animal to do reinforcement learning in an optimal way. However, if different dopamine neurons instead converge onto different regions, that would make different predictions, where different parts of the brain could make different predictions about how to get reward and therefore contribute differently to an animal's ultimate decision. And so I think this is a conceptually very interesting way to think about how reinforcement learning might actually be implemented, the idea that maybe different parts of the brain are predicting different rewards, and also the idea that heterogeneity could still, in the dopamine system, could still be useful for reinforcement learning under this model. And so what this predicts, and again, is something that could be really interesting to investigate from the perspective of the connectome, is that whatever circuit diagram is doing the temporal derivative of these value inputs from the striatum and pallidum is repeated across all the dopamine neurons. And the difference is just that the different dopamine neurons are doing this computation from different inputs rather than from the convergence of all of the striatal or pallidal inputs. And so this is something that I think would come out very naturally from analyzing the input mapping from the striatum to the VTA from an anatomical perspective. And then I want to end by touching on something that we were asked to speak about, which is the one versus many samples. Well, I just want to say that I hope that I've made clear that I think that a single sample would be really valuable in refining and constraining neural circuit models of reinforcement learning. Ultimately, these models need to be pinned to the anatomy, and we just don't have the resolution of understanding of the anatomy yet to really understand how these computations are being performed and even what computations exactly are being performed, as I was saying on the last slide with this question of heterogeneity. And then finally, multiple samples. Well, this is more speculative since we haven't yet analyzed one sample, but it seems to me very likely that comparing across disease states and also across sexes would be very fascinating and could possibly open up multiple fields as that data becomes available. So I think it will be very interesting to know how the connectome differs across these conditions. Thank you very much.